scientist and professor of neuroscience at McLean Hospital, and Dr. Jeffrey Schweitzer, who's also a co-investigator on the surgical side. And this collaborative effort was really designed to try to develop a cell-based therapeutic for Parkinson's, and culminated in a first look at the application of the technology in a paper that came out this past spring, or actually now about a year ago, which was a report in the New England Journal of this journey. So I'd like to tell you a little bit more in depth about this journey and this line of research, and then perhaps give a highlight of some of the work that's ongoing. As you know, in Parkinson's, we have from a cell therapeutic standpoint, the advantage of thinking about a prominent focal network or circuit that is affected in terms of neurodegeneration, recognizing that this network has diverse projections, and of course, this is really a disease that impacts the entire nervous system. The concept here is that if we were able to deliver a cell therapeutic that would replace dopamine and the loss of dopaminergic cells in the nigrostriatal pathway, that we would perhaps be able to alleviate, ameliorate, or even improve some of the symptoms that a patient was experiencing by virtue of the loss of these cells. And of course, this would require us to really produce and obtain a population of cells that we could implant that would survive once implanted. We would also want a cell population that did not have a proliferative potential. That would be a key element. Let's go. We need to accurately deliver the cells to the appropriate target. That's a key part of the technology. And then once the cells are delivered, one would anticipate that an integration process to this degenerated circuit, the formation of synapses, and then of course, appropriate dopamine release and regulation, despite the absence of the normal synaptic input that may have occurred. All of these things would have to occur for this to be a successful approach. We all know that as a neurodegenerative disorder, the concept of Parkinson's is one that as we age, that neurodegeneration is occurring. If we consider intervening at a certain phase of this process, can we either arrest the process, or with a cell replacement therapy, could we have some flattening of that curve, or perhaps even some upsloping in terms of the amount of neuronal preservation at the site of where neurodegeneration is occurring? Over the years, this cell therapeutic replacement concept has been explored by many laboratories. It's been an intriguing concept, and papers as early as 25 years ago utilized a variety of different cell sources to try to accomplish a similar concept. And a number of different strategies and cells were recommended. Adrenal medulla was tried, xenotransplantation was tried, including porcine fetal grafts, fetal midbrain grafts were tried. These were probably most prominently developed in the fetal cell transplantation domain. This involved dissection of ventral mesencephalic tissue, and then bulk preparation on the spot and delivery of cells that involved a fresh or hibernated tissue and homogenized into a small cell suspension. And then, of course, there was immunosuppression that followed. Now, this approach was assessed in a number of different studies, but ultimately, these open label studies, which gave some promise and then gave way to randomized clinical trials, these controlled clinical trials gave disappointing results. While there were anecdotally some patients who appeared to improve upon delivery of this homogenized cell product, overall the benefit was not demonstrated statistically that there was an improvement over a sham surgery control. And furthermore, one of the challenges of this was graft-induced dyskinesias. And this side effect actually made it so that some patients had a worse outcome in terms of the delivery of the cell product than might have otherwise been. As further research has gone on and many follow-up studies in those originally transplanted patients, one of the concepts that's come up is that the challenge of this approach was that really it's a very heterogeneous cell product taken and no two donor products were alike. There was also variation in the presence of serotonergic neurons versus dopaminergic neurons. And in follow-up studies, a number of reports have indicated that really it's likely that some combination of an imbalance of serotonergic signaling and release and dysregulated dopamine release was the challenge with this approach. And some of the follow-on studies that have been published many years later have confirmed that on imaging-based studies, particularly highlighting again this serotonergic imbalance that was a potential etiology. Yet at the same time, some patients seem to have a very sustained benefit. These anecdotal reports, even as late as 8 to 18 years later, patients who were completely off dopaminergic medication and had positive PET imaging that suggested long-term graft survival. So while there are these ethical, practical, and biological limitations to some of the fetal cell transplantation work, newer approaches give promise for other cell sources that actually might be used for a personalized cell therapy approach. One that's intrigued us, of course, and I'll talk a bit about today, is the iPS cell source, but embryonic stem cells, adult stem cells, these have all been explored and are under consideration. The iPSC story was revolutionized by the Yanomaka factors and the concept of being able to take exogenous factors, in this case, proteins OCT3/4, SOX2, KLF4, and c-MYC, expose those to somatic cells and have those somatic cells convert over a period of time to induce pluripotent stem cells, iPSC-derived dopaminergic precursor production to follow. And so the therapy that we've given consideration to is this idea of a personalized cell therapy. It's taking the patient's skin cells, reprogramming those skin cells to iPSCs, and then doing an in vitro differentiation step to make dopamine-producing neurons, and then following this with transplantation. Few questions are key in this concept. Can we do this reprogramming step with efficiency? Can we eliminate undifferentiated cells? Undifferentiated cells could potentially be a significant problem if they were to grow in a proliferative manner after transplantation, forming a tumor. Can the cell product be effective once delivered? The integration concept, forming synapses, regulated dopamine release. Can we improve the survival of the transplanted cells? It's a pretty harsh step to take a cell from a cell culture dish or a frozen vial and then implant it in vivo and then ask it to survive the shock of the lack of oxygen in the initial transition phase, et cetera. And then finally, is there an advantage to an autologous approach where we take the patient's own cells, and this might be from a patient who phenotypically has Parkinson's disease, if we take their skin cells and produce them, are there advantages over using an allogeneic product? The latter requiring immunosuppression typically, but conceptually, potentially with a personalized approach or an autologous approach, the opportunity to avoid immunosuppression. So to address these questions, our group has tackled a few different key innovations. One is the using a technique called metabolic reprogramming, which I'll highlight. Cell culturing techniques, which are designed to produce an efficient differentiation step. Quercetin treatment to eliminate pluripotent cells. And then also a novel surgical transplantation technique to try to deliver these cells in a way that allows them to get past the actual transition step. And again, the autologous versus allogeneic question. This is a question that's being explored by laboratories and groups across the country at this time. We have actually also just this in the past 6 months, there's been an allogeneic trial that's been approved by the FDA, and that's moving forward. And then autologous is the approach that we're working on. And when I say approved by the FDA, approved for a phase 1 study. So let me talk about the first innovation that we tried to tackle. One is to produce cells in a more efficient manner. It turns out that the conversion of somatic cells to an iPSC state is non-trivial. We, through Dr. Kim's expertise and research program, has developed a combination of not only the 4 Yanomaka factors, but also 2 key microRNAs, miR302S and miR200C, which allow for a better programming of the cells from the somatic skin cell state to the iPSC state. And this improves efficiency, it lowers the mutational burden, and it reduces the cancer-causing loss of genomic integrity. And it creates really a better phenotypic profile. So this metabolic reprogramming step is one that actually, by combining these Yanomaka factors with these microRNAs, pushes the production of cells in a way that increases the efficiency. And I believe I have some data here to show you, which is if we take the 4 Yanomaka factors and then we add in these microRNAs which inhibit the SIRT2 pathway, this allows for a significant increase, you can see here, in the colony number of iPSC cells that are produced. So the best combinations, this allows for the survival of the cells in a kind of concept of lower oxygen consumption, increased extracellular acidification, and this metabolic reprogramming. You can deliver these factors with viruses or episomally or even with proteins. Obviously, the microRNAs require nucleic acid delivery. We use an episomal approach in this strategy. So it's a 2 plasmid approach. So in this case, cells, this would be a fibroblast outgrowth. A single transfection is done by electroporation. Then the cells are grown out over a period of about a month and then picked up as iPSC colonies and expanded. And we talked a little bit about the different options for reprogramming. Some individuals have used viral integration strategies. We've elected to not use that, preferring to have minimal effect on the genome. So we've chosen an episomal based approach. We're also actively exploring mRNA-based, akin to the COVID Moderna vaccine, mRNA-based approaches for delivering of these factors as well. The success rate for using an episomal approach is substantial. It gives a good quality product. And so we are currently assessing in a number of patients how efficiently we can do this. And thus far, we've been able to convert the somatic cells to iPSC state with a acceptable level of genomic integrity in a high percentage of patients. Now, let's get back to safety just for a second. This process, the manipulation of a somatic cell to bring it to the induced pluripotent stem cell state, does bring it with it the risk of genomic alterations at the time of this conversion. We do have a rigorous approach to characterizing the iPS cells once they're produced. This was in part mandated by the FDA when we went to perform treatment on our first patient. And this involves characterizing genomic integrity and then also detailed sequencing analysis to show that the presence of any type of non-synonymous somatic mutations was low. So if you look at in the literature, it's certain long-term cultured stem cell lines, you'll find that they actually have a significant accumulation of somatic mutations. This is common with any cell cultured product that's passaged many, many times. Our lines were characterized to have a very minimal level of any type of somatic gene alteration, and specifically to exclude any kind of tier 1 cancer associated genes as defined in the cosmic database. So the idea here was make sure that after the transformation has occurred, the genomic integrity is high and no mutations are present. Now, when you implant or transplant an undifferentiated or a cell that is deriving from an undifferentiated cell, in this case the iPSC cell, we move those cells out to create a product which is essentially a differentiated dopaminergic precursor. So step one is harvest the skin cell through a biopsy. Take about 30 days to move that to an iPSC state, then grow up that colony, characterize it genomically, make sure that there are no genetic mutations or alterations, then move it from the iPSC state out to the dopaminergic neuron state. Once it's out of the dopaminergic neuron state, it's a candidate for implantation. So that process takes about 28 days, and that's we use standard production techniques to bring the cell from the iPSC state to the dopaminergic neuron state. However, along the way, the cells are moving from that undifferentiated state to that more differentiated state. So we have a choice as investigators of when to harvest those dopaminergic precursors and implant them into the patient. And in this case, if you take cells that are just at day 0, if you take frank iPSCs and implant them, you will form teratomas in 100% of animals in preclinical studies. If you move out to day 14, where you're about halfway differentiated, so to speak, from a time basis, you still have significant precursor lesion formation. These are called rosettes, and you can see those after implantation. Now, if you move out even further to day 21 or day 28, it turns out that you can actually eliminate the undifferentiated cells and no rosettes form and no tumors form. But there's a strategy that we've utilized, which is to inhibit a survivin pathway, which allows us these undifferentiated cells to survive by using a quercetin treatment. So if we take quercetin and treat our cell product with quercetin, what we find is that we no longer have rosette formation and we no longer have teratomas that form in mice, which again is a key critical data safety element. And what you can see here is that day 14 cells, which previously I showed you without quercetin treatment, formed both teratomas and rosettes. Now, actually, with quercetin treatment, we don't see that type of formation any longer. And then day 28 cells are even safer still. So for our study, by using quercetin and the combination of this treatment, we've been able to create a product which we feel has extremely low undifferentiation proliferative growth potential once implanted. And if you look at just some of the statistical calculations, whereas you might have without quercetin treatment, you may have in a 10 million cell product, 10 million number of cell product delivery, you may have as high as at day 14, 3,000 undifferentiated or less differentiated cells. With quercetin treatment, we can knock that down to almost no cells. And then as we proceed to day 21 or day 28, we have theoretically even less than a single cell that would have that type of proliferative potential. So we've incorporated this into our differentiation protocol. We've also tried to develop some optimized protocols for producing the cells. It turns out that as with any cell product, this requires a careful cell culturing to do it at scale. Part of this was to develop a spotting technique, which allows for the cells to be placed in such a way on the cell culture dish in spots throughout the dish. And this expands both their viability, decreases their detachment, and promotes their growth in a way that is clinically viable in terms of scale. And so this is what the total protocol differentiation looks like from day 0, exposure to first floor plate induction, neuroprogenitor induction, and then dopaminergic precursors by day 28. We, of course, characterize these cell products phenotypically to make sure that they do all the things that we want them to do in terms of both immunohistochemical markers and then cell productions and then electrophysiologically responsive dopaminergic release. We of course were required by the FDA to show in preclinical models that there was correction. We used an induced 6-OHDA lesioned rat model for for Parkinson's or dopaminergic circuit degeneration. And what we find that if we use day 14 differentiated cells, day 21 differentiated cells, or day 28 differentiated cells, we do see correction by about 4 months in the animals in terms of a rotational behavior that's characteristic of this lesioning model. And we see obvious vigorous engraftment with Th positive dopamine producing neurons in place. So about in an animal model, 4 months to correction of that abnormal rotational behavior. And then if we do it, we've done it in many different types of rats and shown a similar pattern of correction on, this was a rotation assay again, by about 20 to 24 weeks, we start to see again that positive signal of correction. A time period which we feel is required for allowing the graft to integrate into the local circuit and begin to produce regulated dopamine. We can do this with either fresh cells or cryopreserved cells. In our first patient, we used fresh cells, but we've now tested cryopreserved cells and found that we also have good preservation of viability. And the cryopreserved cells, of course, make it considerably easier to envision moving along to a phase 1 clinical trial. In terms, again, of graft, the histology suggests that once the graft is in place, we, again, have all the appropriate markers for a differentiated dopamine-producing cell product. Another key innovation was the transition from the cell culture dish to the actual implantation. Many studies have been done where the cells have been delivered in a simple bolus. So a needle is passed to a target location, and then a small bolus of the cell suspension is delivered. This basically expands from the tip of the needle in a sphere. And as you can see, when we look at this histologically, we get disruption of the normal tissue. We also have loss in the center of the graft, probably related to the fact that oxygen and other nutrients have a difficult time initially penetrating into a ball of cells that is simply delivered. So one of our investigators, Dr. Jeffrey Schweitzer, developed a technique called columnar injection. And in this technique, what happens is a needle is passed to a core depth. And then as the needle is withdrawn through a specially designed mechanical device, it essentially deposits cells along the tract in a column. Now, this actually improves the surface area to volume ratio, increasing the surface area to volume ratio so that nutrients, as they penetrate into the column, don't have as far to go to get to the center of where the cell product is. And we think this does facilitate the initial survival of the cells upon transplantation. We know that the majority of cells actually don't survive, even with these types of techniques. So any improvement in the actual cell survival is important because, again, the vast majority of cells don't survive in any approach. So this is, again, what the columnar injection technique looks like. As compared to the bolus just delivers the sphere at the tip of the needle, where the columnar technique actually draws the cells out into a column. And then we found on our studies that this gave an improvement of the number of surviving neurons at 3 months post-implantation in vivo. And this was something that was published a couple of years back. So let me just transition to describing our workflow for a single patient study that we embarked upon. And I realize I'm maybe pushing on my time here, so I'll try to go a little faster, if that's okay. This was something that we started several years ago, went through a number of iterations in our discussions with the FDA. We performed an initial patient with a left-sided surgery in September of 2017. This was our targeting software that showed where we implanted the cells at the time of surgery. We did 3 columnar targets, a total of 4 million cells, using a single cortical entry site. The FDA mandated that we separate the 2 sides by 6 months. So we did one side in late 2017, the other side in early 2018 in this patient. And this is our team in the operating room here at Mass General, the columnar delivery device. And we were also mandated by the FDA to confirm our target with intraoperative CT, which we did. And this shows that we had a good delivery right to where we wanted. So what was the short and long-term follow-up on this patient who was delivered. You can see here that just 24 hours after the right side surgery, you can see the 3 small dots, which are the tracks of delivery on the right side. Now, here, 18 months and 2 years after the 2 sides were treated, you see the 3 tracks, you see a small area of flare positivity right around the delivery site. It turns out if you get into the literature, that flare positivity is seen in many cell delivery approaches. And it may be any number of aspects related to conceivably local microinflammation, the actual integrative process for synapse formation. What we saw in PET scanning was in this particular patient, a very modest increase on the order of 10 to 15% on the right side and 2 to 10% on the left side of increase in cell signal. We started with a very low dose on this patient, who was our first patient. It's primarily a safety study, but we were encouraged to see some signal that was delivered. And then this is, again, the PET scan. So 2017 scans, 2018 scans, 2019 scans. And the nadir for this patient was his late 2017 scan. And then we can see some increase in signal that had occurred at those lower levels that I described on these later PET scans. We wanted to make sure that our theory that an autologous approach would be a viable one from the immune suppression standpoint. This patient did not receive any immune suppression. We created a humanized mouse model and studied this patient's immune system in relation to their graft. And what we found, and I'll just summarize this slide pretty quickly, which is that this patient's cells implanted into a mouse that was humanized with his own immune system showed graft survival and no rejection. Whereas these same cells implanted into an allogeneic donor humanized mouse showed rejection. So the theory that we had espoused of immune protection of the graft by an autologous approach seemed to bear out, and this was shown in this experiment. And this again was a highlight of that experiment. And histologically, I won't go through this in detail, but we were able to show that those cells did persist in the autologous approach. Okay, so clinical outcomes. The patient has remained on levodopa and had modest alterations in UPDRS scoring. The patient had improved sort of non-motor quality of life symptoms, symptom scales. And this was kind of curious to us, and I can go into some detail perhaps in the question and answer period about what some of those improvements that the patient reported were. Just to kind of put a little real world face on this is the patient pre-surgery. He's an avid sportsman. He's here in the white. He's trying to basically do some skiing. And he has an assistant here who's with him to kind of watch him. As you can see, the fluidity of movement is modest. He has his baseline tremor that exists. And so 2 years post-implantation, the patient has still been able to enjoy his activities. He feels that some of the subjective fluidity of movement is improved, and this is kind of the way he's able to sort of live his life at this time. We are cognizant, obviously, that a single patient proof of concept study is limited in what it can tell us. Parkinson's is so variable within a 24-hour period in terms of how a patient experiences their symptoms that no single patient, of course, will give us any data sufficient to make any kind of conceptual notion of outcomes. So we have expanded our approach. What we feel like we've learned so far is that we can produce autologous iPSC-based dopaminergic neurons in a safe manner, and we can get them to a clinical grade sufficient for implantation from a safety perspective. We've tried this in a single patient, and we've learned a lot about the process of producing the cells. We have some preliminary data on graft survival. And at this juncture, we feel like we're really ready to move to next steps. So we're building out collaborative approaches to developing a phase 1 trial. We have a considerable amount of work going on to further optimize the production phase and strategic approaches. The report received a bit of commentary, and you can read some of this in Cell Stem Cell, and highlighting the differences between the autologous and the allogeneic approach. And I think what I'll do is stop there with appreciation to everyone, and open up for any questions that you might have on what we've learned thus far.